I'm really excited this afternoon to be able to moderate a panel discussion with several of our industry partners. We've got 6 folks here joining us from different companies, all developing therapies for FA. And so I'm going to ask those folks to come on up. We are going to need a few extra mics, Jamie. If you can just grab them and bring them back up, that'll be great. And while those folks are coming up, I will just give a little bit of background for you. So if you could... Oh, I lost my mic. There we go. And I lost the slide advancer, so maybe just move forward for me. That'd be great. One more. Terrific. So this is the treatment pipeline. You can sit in any order. I'll pick on you no matter where you sit. So this is the treatment pipeline, and I showed this earlier today, and we're going to talk about several of the programs, 6 of the programs on the pipeline. If you go to the next slide, I'm going to zoom in on what's at the top of the pipeline. Thanks, Jamie. And so the top of the pipeline kind of shows you the process of drug development, going from early discovery all the way through having approved treatment. So starting in the lab, working our way through clinical trials, and then to approved treatments. And we draw this as a straight line. It is not. It involves a lot of back and forth before you get all the way through it. And so you'll hear a little bit of that today. When you look down the left side of this chart, we're talking about different ways in which we are trying to approach therapies. And so we start if we start at the bottom, working our way up, we're starting with the gene itself that causes FA and the mutation. And then going towards the middle, we're focused on things like the protein, and then the mitochondria, and even symptoms. And so when we think about developing therapies, we look at all of those things, from the symptoms all the way down to the gene mutation. And the goal is to have multiple different therapeutic approaches at all of those points. And that's kind of how you get to this treatment pipeline. We know we're going to need lots of different therapies to ultimately achieve our goal of treat and cure FA. And the other thing we're trying to do is bring, like I said, bring new things in here, but also help move them along quickly and efficiently. I just wanted to mention that earlier this year, many of the groups working with us to develop therapies gave webinars. All of these webinars are available on our YouTube channel. You can also access them through our website, through the pipeline page when you look to learn more about each of the programs. We don't have the time today to go like deep into all the programs, but there's a lot of information on that pipeline page with these videos that are available to give you more information. And then these are the programs that we're going to cover today. So we're going to talk about omaveloxolone, we're going to talk about vatiquinone, nomlabofus, DT216P2, and then 2 new programs, the 2 gene therapy programs, LX2006 and SGT212. And it is a tongue twister. I don't make up these names. Nobody asked me. I had to come up with something much easier, A, B, C, D. But these are the programs we're going to cover today. And so really grateful to our panelists who volunteered to join us and share more about their programs. So with that, we can cut the slides, and we're going to go into more of a conversation. So I'm going to ask each of you to maybe introduce yourselves, tell us a little bit about the program that you're developing, and kind of what stage of development it's at. And Nicholas, I'm going to ask you to start off.
Okay, thanks. Thank you. So I'm Nicholas Folschweiler. I'm the clinical lead for omaveloxolone at Biogen. So thank you all for being here today, and thanks for the invitation. Really, it's a pleasure to be here. It's an honor. I had the chance as well to do the ride yesterday, and my voice has still not recovered from that. So on behalf of Biogen and the Biogen team, I would like to thank FARA for the leadership and for creating a space like this where we have the science, we have you, the patients, we have advocacy, and we have the industry, so the medications that come together to progress the knowledge on FA. So Biogen actually is been around for quite some time. Biogen is a place where we develop medications that makes a big difference to the patient where there is a big need at the end, and OMAV has its full place at Biogen. Biogen actually, and the story of omaveloxolone started quite some time ago with the collaboration with FARA actually, and on the MOXIE clinical trial program. You may have heard of MOXIE, the name. MOXIE was the pivotal phase 3 study that led to the registration of omaveloxolone in patients aged 16 and above. So it was based on 3 years data that were mentioned a little bit earlier today. FDA approved this 3 years data 2.5 years ago, 2023. And actually, I'm very pleased to let you know that last week we disclosed at 2 congresses 5 years data of omaveloxolone, and the results of this 5 years data were consistent with what was observed with the 3 years data 2.5 years ago. Now, a big piece that is taking a lot of efforts these days is the BRAVE study, so the phase 3 pivotal study in the children and young adolescents, so from the age of 2 to less than 16 years of age. So the study actually started in June this year. So we had our first center opened in Virginia, in Norfolk, yeah. And we are now opening additional sites. So the CHOP site opened in August. And we're adding sites in the US. So there will be 6 sites in the US opening in the coming days or weeks. So you can still look at clinicaltrial.gov, which will be updated regularly with the new sites. There will be also sites outside of the US for BRAVE. So yeah, so BRAVE is really kicking up now, and we are starting this study. The last piece that I want to talk about is still the collaboration with FARA on the UNIFY natural history study that was mentioned earlier today. So the natural history studies are very important. So UNIFY combines the patients from the FACOMS dataset, FACOM natural history study, and the EFACS natural history study. And through the combination of these 2 studies, we started 2 real-world evidence studies. So one is the PASS, post-authorization safety study, that will investigate the safety of omaveloxolone in the long-term use. And the second study that we start, that was also mentioned a little bit earlier today, is a pregnancy and lactation registry study, which will document the use of OMAV in patients who are pregnant and or lactating. So that's in essence our program with omaveloxolone these days. Thank you.
Nicholas, thank you. I have a few follow-up questions for you. Maybe first, you know, omaveloxolone, it's kind of in this red part of the pipeline, so it's focused on improving mitochondrial function, right? And so maybe just a quick overview on kind of what omaveloxolone is for some of the families maybe who are new to the community and aren't as familiar with it.
Yeah. So omaveloxolone, so it's a chemical, it's a NRF2 activator. So the idea of omaveloxolone is to interfere with the metabolic pathway in the mitochondria and to give back some energy to the mitochondria by improving its functionality. So that's the way omaveloxolone is working.
Great. Yeah, thank you. You mentioned that the BRAVE study, the pediatric study, is just starting up and getting going, and it's my understanding there are going to be sites around the world eventually, in Europe and Australia and South America. But can you tell us a little bit more about the study itself? Like, what age ranges are being enrolled, what might be involved in participation if people are have children with FA, they're thinking about clinical trials, and what are some of the primary goals and objectives of the BRAVE study?
That's a lot of questions.
It is a lot. Well,
so the study, yes, so it's a global study. So actually it's a very big study. It's 255 patients, aged from 2 to less than 16 years of age. So we split, of course, in the study, the age range 2 to 6 and 7 to less than 16. We will have both ambulatory and non-ambulatory patients from everywhere, nearly everywhere in the world. So it's a study where we plan to enroll patients in more than 30 sites across, I think, 14 countries. So a very large study. The primary endpoint is USS. So USS, Upright Stability Scale, is a subset of the mFAR score that you may be familiar with, because USS is a key driver for FA in this population. So it's different from the adult population. So we believe that USS is the right endpoint to assess, and that was discussed with Patients Association, with the regulators, and with the key experts in the field. So we all agreed that USS was the right endpoint for that study. And briefly, so the study lasts, so there's 1 year of placebo-controlled phase, so that's the part 1, and the randomization is 2 to 1, so you have double the chance to be on omaveloxolone compared to placebo. And at the end of this part 1, so at the end of this 1 year study, patients will all move to an open label phase where everyone will get OMAV for at least 2 years, or for 2 years.
thank you. I think you got them all. That was good. So, FARA, we often get questions from community members about OMAV, and would it impact other symptoms? So you talked about how it, improved mFARS function, and you're looking at in the BRAVE study its ability to maybe maintain or improve upright stability in children. But what about other symptoms like cardiac? Do we know or are we looking at in the long term what other potential benefits or side effects there might be to OMAV?
Yeah, for the symptoms in general, so including cardiac, so there's the PASS study that I mentioned that was put in place, and that has started, post-authorization safety study. So this safety will look in a long-term setting on the safety aspect of omaveloxolone in the indicated population. So the cardiologic issue will be monitored as well as part of the overall safety monitoring. Yes.
Thank you. One last question for you for now. For those folks in the audience who are over 16, and if they are also new and they haven't heard about OMAV or Skyclarys or Biogen, where do they go for information? Or if folks are having trouble accessing OMAV, are there programs in place that you can refer them to?
So the first thing is to talk to your doctor. That should always be the first point of contact. Talk to your doctor. The second is for people above the age of 16. So then Biogen has a program which is called REACH that would help your physician or your doctor to fill the forms and to understand whether you will get coverage, insurance coverage for the medication. So that's one part. And the second part that could help you as well is still at Biogen. There's a program that is called Family Access Managers that can guide you through the process of getting access to omaveloxolone.
Thank you. I'll let you off the hook for a few minutes. John, you're up next.
All right. Hi, my name's Jonathan Cherry. I'm the global project lead for vatiquinone, and I'm here to represent PTC. First off, I'd like to say thank you for all having me here. This community is amazing, and I'm honored to be even a small part of it. So I love coming to all of these meetings and talking with you, and if there's any questions, find me afterwards, and I'm happy to chat. PTC is a small pharma working on rare disease. We've been involved in a number of approvals. PTC worked with Roche to get Evrysdi approved for spinal muscular atrophy. And just this year and last year, we had very exciting news on Kability, which is a gene therapy to treat children with a very severe disorder called AADC deficiency. And Sephience was just approved for kids and adults with PKU. Again, a disease that makes it very difficult to digest protein and creates quality of life challenges for those patients. I'm here to talk about vatiquinone. Vatiquinone is a 15-LO inhibitor. So 15-LO is 15-lipoxygenase. Very scary word. Let's just call it 15-LO. It's easy to understand. And what it does is it works in the mitochondrial disease pathway. So if you guys all remember high school biology and the powerhouse of the cell, when you have FA, you lack enough frataxin in order to properly generate iron-sulfur clusters, which help you create that energy, create that powerhouse. And some of the byproducts when that breaks down is that you get oxidative stress, oxidative species that can cause damage to DNA and cause damage to lipids. It also creates this process of iron dislocalization. When that happens, your cells, in a healthy context, will take those, and through 15-LO, they'll take those reactive oxygen species, those toxic species, put them in lipids, and then those get detoxified, and it's fine. But when you have a chronic disorder like FA, the mechanism by which you can turn those toxic lipids and detoxify those gets overwhelmed. So in order to prevent that buildup, we inhibit 15-LO, and we don't get those toxic species. In FA, what happens is those toxic species get into the mitochondria, and they get into the lipid membranes, and then they kill the cells. So really, that's why you lose the sensory neurons, where your cardiomyocytes die, and why you have the problems in the dentate nucleus. So all of those cells die off, and by inhibiting 15-LO, you can preserve those cells, and although they're not necessarily as healthy as a normal cell would be, they can still function, and that still allows you to be able to minimize the slow progression of disease. So where our project currently stands, I think many of you are aware that we did get some bad news from the FDA recently. They told us that our approval was not accepted, and really the focus is, and I'll say I'm horribly disappointed, as I'm sure many of you in the room are as well. The FDA looked at our data, and when they looked at what our primary endpoint was in our study, which is MOVE-FA, they saw that we did not reach the statistical hurdle for approval based on mFARS, which was our primary endpoint. Our package was based on data we saw in upright stability, which is a component of mFARS that focuses on balance and stance. And because MOVE-FA was a study designed to look at, it was designed to look at overall function, but what happened in this study is we enrolled very young patients. We had patients between 7 and 21, and the majority of our patients had age of onset under the age of 10, and the average patient in our study was 14 years old. So we had a very young, early onset population, and as was found using, again, the natural history, the FACOMS natural history done with Dave Lynch and Christian Rummy, and I think, Jen, you're on that paper too. We see that younger patients with early onset really show progression primarily in that upright stability subscale. And those other things, upper and lower limb, while there is dysfunction, it is not as uniform or as reproducible from patient to patient, and that brought noise into our result, which is why we did not reach that statistical hurdle on the mFARS.
John, thank you. First, let me thank you and PTC for being here even after getting bad news from the FDA and staying committed to the Friedreich ataxia community and developing vatiquinone. That doesn't always happen, right? When research doesn't turn out the way you want it, when you don't get the result that you needed, sometimes programs can't continue, and folks do leave. And so it means a whole lot that PTC is staying committed to all of us. So we are really grateful for that. I'm going to try and maybe connect a few more dots in some of what the overview that you gave. So you talked about MOVE-FA. For those who don't know, MOVE-FA was a double-blind, randomized, placebo-controlled trial. It was global. It enrolled 134? 149 people with FA, majority children. And as John mentioned, when you design clinical trials, you try and predict before you do the study what your result's going to be. And so when you hear primary endpoint, that's what you think the result's going to be. And so going into the study, it was thought that we'd see a benefit on mFARS. And unfortunately, that benefit that was seen on mFARS did not reach statistical significance, but a subset, a subscore within mFARS, the upright stability, did reach statistical significance. I got it?
Yeah, that's correct. And I think it, to go a little deeper, I was trying to stay on the surface. But in mFARS, we did see a benefit, and it was a clinically significant benefit. But because of that noise, because of that variability, mFARS itself didn't show the statistical significance. But upright stability as a subcomponent and as a standalone was both clinically meaningful and statistically significant, and that was the basis for our approach to the FDA.
And assuming also that the drug was safe and well tolerated.
Correct, yes.
So I think you started to go over this, but so again, for folks who are new, after the trial, PTC submitted an application, what's called a new drug application, to the FDA. And that in that new drug application, you're asking FDA, here's the evidence, here's the trial results, here's the safety, here's the efficacy. You can review all of this and hopefully find that the potential the benefits outweigh the risks and that this should be a medicine that gets approved. In this case, when FDA completed that review process, they said, no, we don't think the drug should be approved. Can you help folks understand? I know we don't really understand, but what did the FDA at least say in the no? Like, what reasoning did they provide?
That's fair, and I think I'm going to have you sit in my spot, because you're answering my question. You're doing my story better than I am. I love it. So what, essentially, when they send you a no, they send you what is called a complete response letter, or a CRL. So they document all of their concerns with the data package and what they had what worked and what didn't work for them. What it really boiled down to was that, by definition, in a clinical trial, you have to meet that endpoint that you predicted was going to be positive. And you design your trial and you enroll a certain number of patients to meet that in a way that is robust enough to give people confidence that it worked. Because we missed that, we missed the primary endpoint, and because the FDA really wanted to stick to the definition of what the guidances are, they were very conservative, and they did not approve based on the primary endpoint, despite the fact that we did see the benefit from upright stability. So really, this boiled down to us not being able to hit the primary endpoint, despite being able to show that we had, again, robust and reproducible effect on upright stability.
So what's next? What's the options going forward?
Well, I'm still here.
I know, that's why. All right, where are we going?
We're not done yet. I think when you get a CRL, when you get a complete response letter, you have 3 months in order to re-engage the FDA, discuss the points within the letter, and try to come together for a path forward. We've been working very closely to reanalyze data, identify new things that can hopefully help them understand and see our perspective on how upright stability really was the most meaningful. And we're going to re-engage with the FDA, and they have a lot of different mechanisms. I will say that every pathway is open to us, every option is open to us. We're going to do whatever it takes to keep this moving forward. And we're going to have to, the first step is a conversation with them to agree to those steps. And that's called a Type A meeting, and we're going to submit that soon in order to align with them on what is going to be acceptable for them, for us to move forward with an approval. And like I said, everything's on the table, and we're just going to give them our perspective on this and make a proposal and see what they have to say.
And has there been an opportunity for the community perspective to be a part of that?
Unfortunately, as part of the review process, there's sometimes a thing called an advisory committee or an AdCom, and in those meetings, the community is invited to speak. There was no AdCom for our submission, which was unfortunate. So the community voice didn't really come through. That being said, I know that FARA is very strong. Again, between Jen and Ron, we know that they're not quiet. They fight for you, and they're letting people know. But again, this is something where advocacy comes from you, and this is why the section today was so important, where it's your voice that is strong, and you can advocate for yourself. My job is to create the data package that can, with that advocacy, allow us to get that approval.
For those who don't know, Chandra, raise your hand. Chandra led an effort after we got the CRL from FDA. She reached out to us and said, we need to share the community voice, and I want to lead this effort. And so she reached out to about a dozen different community members and asked them to provide testimony in a written format. And that written format she collated that into a whole package and has submitted that. And that came from parents of young children, parents of older individuals with FA, individuals with FA themselves, some who've had experience with vatiquinone, some who have not. But she collated this diverse set of experiences from the community to be representative. And Chandra is a scientist herself, and so she dug into the statistics of the FDA's response and tried to appeal to them both as a patient and as a scientist, and explain why she humbly disagrees with their assessment. And but she submitted all of that to PTC so that when that Type A meeting occurs, PTC will at least have the community voice available and to be represented. So Chandra, thank you for that. And thank you to the people who wrote letters for Chandra. Rusty, I'm going to ask you to tell us a little bit about Nome Labofus, about yourself, about Larimar.
Okay, thank you, Jen. And I want to echo the other folks on stage here by thanking FARA first for having this event. This is, I go to a lot of these events. This is my second favorite event of the year. And it's really an honor to be part of this panel. I have to say, the FA soiree in Oklahoma City is my favorite.
All right,
Anyway,
I get it. I can't sing.
Try harder.
You don't want me to.
If you haven't gone, by the way, it really is a treat. You're welcome, Eric. So I'm Rusty Clayton. I'm the Chief Medical Officer of Larimar Therapeutics. We're headquartered in Bala Cynwyd, Pennsylvania, which is 14 miles due east of here. And we're developing a Nome Labofus, by which Jen, good job on pronouncing it. We're going to call it NOMLA for short, though, because it's a lot easier on everybody. As we all know, Friedreich's ataxia occurs because of a deficiency of a protein called frataxin. And so very simply, NOMLA is a frataxin replacement therapy. This was developed by Mark Payne at the University of Indiana. Mark's a cardiologist with a keen interest in FA, and he found a way to take the frataxin protein with its partner protein, a mitochondrial targeting sequence, and he attached a small cell-penetrating peptide to the end of it, and this is what Nome Labofus is. It's injected once a day under the skin, pretty much like insulin was injected for people with diabetes, and it's designed to increase frataxin levels in people with Friedreich's ataxia. We're currently a clinical stage company, stage 2. We have an ongoing open label study. It used to be called an open label extension study for people who had participated in a prior study, But we've recently opened it up to people who hadn't participated in a prior study and give them the opportunity to participate in our clinical development program as well. And this was done because when we designed our phase 3 study, which will be coming live shortly, we realized there was still a lot of folks with FA who may not be able to participate in the program. Phase 3 studies are funny that way. You've got to be somewhat selective and restrictive. So we decided to call this the open label study and invite other folks into the study. So that's our ongoing study. As I mentioned, we have a phase 3 study that's going to be opening shortly. We just concluded an adolescent study in the spring, and we had a kind of a general announcement of our program just a couple weeks ago. A very detailed press release. It's on the Larimar website, so if you haven't seen it, feel free to go to the Larimar website. Just go to the investor page, pull up the press release, and it gives a summary of our program. So rather than take up time here, just gave you an overview of what we do.
Thanks, Rusty. If I were to kind of give a high-level overview of some of the information that was shared 2 weeks ago, there was information from people who have now been taking NOMLA for 12 months, right, in the open label study, to show kind of how they're doing, their mFARS scores, if there's evidence of clinical benefit, the frataxin levels. I think the frataxin levels were measured in the skin, and there were significant increases in frataxin levels up close to carrier level. So there was a lot of good data shared about the experience of NOMLA going out over 12 months. It's still, there's small numbers of people who've actually been on it that long. There was also other important information that you shared about sort of the safety and adverse events. There were injection site reactions were the most common adverse event, if I recall correctly. And then another adverse event related to anaphylaxis. Can you help folks understand what is anaphylaxis?
Good question, Chen. So anaphylaxis is an allergic reaction. What an allergic reaction is basically is that something in the environment or something that you're exposed to, your immune system sees it, decides it's not part of you, and decides to react against it. This happens to a lot of people in the spring, for example. They have an allergic reaction when there's pollen in the air because the body doesn't like the pollen, and so they react. The difference between a runny nose from an allergic reaction and anaphylaxis is a lot more serious. Anaphylaxis is a serious and potentially life-threatening complication or allergic event, if you will. It happens in response to environment, food allergies, and in fact, anaphylaxis induced from drug and protein therapy is the most common reason why people develop anaphylaxis. So just to repeat, very serious life-threatening allergic reaction. It sounds scary because it is scary. It's scary and unpleasant to experience. It's very scary to watch when somebody that you care for is having a case of anaphylaxis. And to be honest, it's even scary for doctors, and particular neurologists who don't deal with anaphylaxis. And it's one of the things that's troubling our investigators right now is this anaphylaxis. So that's the bad news. The good news is that the anaphylaxis cases that occurred during our study, all of those folks responded very well to the standard treatment of an epinephrine or adrenaline injection. This is the treatment for anaphylaxis, and almost all of the time, people respond to that treatment very well. In other words, they return to their usual state of health very quickly. And that's what happened with the folks who developed anaphylaxis in our study. They got the epinephrine, and then they returned to their usual state of health. Unfortunately, if you have that type of allergic reaction, we can't keep you in the study. So those folks had to be withdrawn from the study, and we're trying to develop a program for desensitization or something like that, so we can get folks who did have an allergic reaction to come try the drug again once we desensitize their immune system for that. But anyway, that is anaphylaxis in a nutshell. Very serious, and the company is taking it very seriously. And as a result, for example, everybody who enrolls in our study goes home with an epinephrine autoinjector. In fact, they go home with two.
So thank you for helping us understand what the risks are, or what the risk is, and how it can be managed. What is the chance of somebody having anaphylaxis? Do you know?
So it's, sorry, it's an ongoing study, so it's hard for me to say. Right now, and it's a complicated question. Most of the folks who enrolled in our study had been exposed to the drug in a prior study. This is a great setup to have an allergic reaction from anaphylaxis. You get the exposure, there's a period of time where you don't have the exposure, and then when you're re-exposed, that's when you have the allergic reaction. In that setting, which we don't expect to have happen in the real world, we expect people to continue to take their medicine and not have a break of months or years, the rate is somewhere between 10 and 20%, which is high. Other protein therapies that are out there, and most of them, if not all of them, have at least some warning for anaphylaxis. The rate is more around 10% or sometimes less. So it's a little high, but like I said, this is a moving target right now. We don't know what the true rate of the anaphylaxis is. I will say this, though. People have heard, I saw the ears perk up when I said the words life-threatening, and that's good because I said that on purpose. You need to hear that. In actuality, the mortality rate from anaphylaxis from drugs is about 1%. And if you have an epinephrine autoinjector, that rate drops to 0.5%. So the anaphylaxis may happen 1 out of 10 times, 2 out of 10 times, but as long as you have the autoinjector, it is, in fact, a manageable complication.
Thank you for that. You had mentioned that the study is going to continue to enroll people who have not been exposed to the drug previously, and so hopefully that will give us a better understanding of what this risk is over time. But maybe just a question that came to mind as you were explaining kind of the start and stop and how that might make people more susceptible to it. Maybe a clarification. NOMLA is what's called a biologic. Like, we're giving people a protein that's different than some of the small molecules like OMAV or vatiquinone that we talked about. So it's not all medicines that you start and stop that you can have an allergic reaction to, but specifically what's called a biologic or a protein. So maybe just in the last few minutes here and questions going forward, you mentioned that the open label study will continue to enroll people who might not qualify for the phase 3. What's planned for, or what can you say about that and the phase 3 study?
Okay. So first, we're going to finish enrolling the folks who previously participated in a study. They're still waiting to get in. So as long as they want to participate, we're happy to have them. We're also in the midst of recruiting folks who have not participated in a prior study into the open label study. And in fact, we opened a website. I think there's information on the FARA website that helps folks find that link, and you can register for that study if you think you're interested. And by register for that study, I mean you can say, hey, I've got Friedreich's ataxia, I'd like more information. I don't get to see that information, that's a third party, and they'll help guide you to sign up to be screened for that study if in fact you meet eligibility requirements. Along with that, and because of this pause that I mentioned that we saw between, in these patients, we believe the pause increases the risk of the anaphylaxis. And so for that reason, we didn't do a study in younger children under the age of 12 like we did with the adolescents. Instead, those younger children will be folded into the open label study. So there will still be an opportunity for children with FA under the age of 12 to enroll, where we will be looking at the pharmacokinetics, the safety, and frataxin levels in that youngest population. That's the phase 2 study. The phase 3 study will open up later, and this will be a global study. So similar to the BRAVE study, it'll be not just in North America, it'll be in Europe, it'll be in Australia, And we'll be recruiting people as young as 2 years of age eventually, and people up to 40 years of age with Friedreich's ataxia. And this will be a phase 3 study designed to confirm the safety and efficacy of nomlabofusp.
Thanks so much, Rusty. I really appreciate your going over the anaphylaxis risks, the safety, the concerns. This is one of the first times we've had a medication where there are significant risks associated with taking the medication. And so there's a process before you join a clinical trial called informed consent. And this is where you meet with the study team ahead of time, and they explain what's involved in the study and the potential benefits and the potential risks. This is an incredibly important part of the process, and so that's an opportunity to get a lot of questions you may have answered. And all of the information that is known about NOMLA right now is in that informed consent document, and the study teams are knowledgeable and able to kind of help you work through that risk benefit. We also, FARA has a clinical trial guide, and the clinical trial guide in the back kind of takes you through some questions to ask yourself as you go through a study process, and specifically relating to having to weigh risks and benefits. So I just want to make sure that you know that those resources are available to kind of help you make decisions. These are not easy decisions to make. And it's important that you make a decision that's well informed and that's right for you. All right, I'm going to move on to Pratik. Pratik, can you tell us about DT216P2 and design therapeutics?
Sure, I'm Pratik Shah, and I really appreciate a chance to be here. My name is not important, but the important name is for Design Therapeutics. And we are here, I'm here on behalf of a team that is very dedicated to working on a new therapy for Friedreich's ataxia. I would say it's the reason for us to get up in the morning and do what we do. And the reason we named the company Design, actually, if I go back to the beginning of the company, it was 7 years ago when Ron and Jen and us met up to talk about, what could be done. And the company was based on original research that was funded by FARA. And then FARA has been an important early supporter that helped the company in its very early days to get started. So we're very grateful for the support of FARA and for the support of this community in doing the work that we do. We named the company Design because we asked ourselves, what could we design that might be, in theory, the type of medication that one would want to create. And it was out of that desire we realized that the thing that one would love to do is provide endogenous frataxin, which is natural, that everybody in this room already makes. But what if we could get that endogenous frataxin to be produced in all of the cells in an individual in a manner that would restore the cellular function? What if that could be done? And not only we asked ourselves, what if that could be done? What if that could be done with a small molecule? And the reason I say small molecule is because small molecules in general in medicine go into all the cells naturally, they go into all of the organs naturally, and that was the reason to start Design and to create what is now named DT216P2. And so what DT216P2 is a small molecule investigational medication that engages directly and recognizes those long GAA repeats that are sitting in the frataxin gene that sort of act like speed bumps in the gene itself. And what DT216P2 is designed to do is lay over those speed bumps and smooth the path for the machinery in the cells to read the frataxin gene, the endogenous natural frataxin gene, without any gene editing or gene therapy or exogenous deliveries, and restore the expression of natural endogenous frataxin. That is what DT216 is designed to do. Now, we did thanks to many of the people in this room and this community, we did get a chance to try DT216 in a clinical trial in 2023. And we got some very exciting results, but we also had a setback. And the exciting results are, the setback was that the administration was intended to be once weekly, and what we found is that the drug was only sticking around for about 2 days. And so what was supposed to be a multiple dose study turned out to be a series of single doses, a week apart. But despite that very short duration of exposure, we were able to observe an unmistakable increase in endogenous frataxin RNA expression from the endogenous gene in both in the blood cells as well as in cells from affected tissues like skeletal muscle. And that was very exciting. The bad news was that we were not able to increase dose frequency because there was sporadic and infrequent vein inflammation, what we called injection site thrombophlebitis. And that limited us from dosing more frequently or increasing the duration of exposure. And at the time, it wasn't clear if this could be solved. And so we've had our heads down for the last 2+ years, and I'm delighted to say that we were able to create a new formulation of the molecule that we took into the clinic in 2023. And that new formulation is called DT216P2, which is essentially the version 2 of DT216. And I'm also delighted to say that we're back in clinical trials in patients with FA, and we're very excited to see what the results of those would be. It's early days. We have a lot to learn about what DT216P2 is like and what it can do. But given the inherent mechanism and what we've been able to observe with increasing frataxin expression, both RNA and protein, in cells taken from individuals with FA, whether they're blood cells or cells that have been turned into heart cells or neurons, we see this very exciting observation of increasing endogenous natural frataxin. And it is with great hope that we continue our work, and we thank all of you for your support and participation.
Pratik, thank you. I think this is a good example of when I talked about before drug development looking like it's a straight line, but actually we go forward a little bit, and sometimes we have to go back, right? And in this case, we had to go back. They had to reformulate the drug. They had to try and work through some things that they saw in the phase 1 that they knew they wanted to make better. So it's really encouraging that you're back in the clinic. And so I guess that means that kind of the injection site thrombophlebitis that you saw, and maybe some of the issues with the drug staying in the system long enough, those are things that you now think you've been able to address and see that in both the what we call preclinical studies, right, in the animal studies, and then you're now looking at that in, you said, a first-in-human study, right?
Yeah, so what we learned is that in the original formulation, there are these things called excipients, which is the stuff that you put into the solution along with the drug to help make it an appropriate injectable product. What we learned in going back and looking very thoroughly in preclinical studies was that it was the excipient that was a huge factor in causing this particular type of response in the vein inflammation. And so we worked really hard to see if we could find a different way, a different set of excipients to create a new injectable form of DT216. And what we found in all of the preclinical studies is we saw no such issue in all of the preclinical studies that led to our preparation to get back into clinical trials. And then the first portion of our clinical trials was in healthy volunteers. And so, we've completed that work at a level where we believe we've confirmed that all of those issues that we had seen in the previous formulation appear resolved based on the healthy volunteer work, and now, confirmed with some of the early work that we've been able to do in the ongoing study thus far. So we think that that particular issue, we believe, has been resolved in a way that wouldn't really hold us up from continued exploration of DT216 to really see what its potential is in the clinic.
The study that's ongoing now is called Restore FA, correct?
That's right.
Yeah, and the goals of the Restore FA, like you said, are to look at the safety and the dose. And I believe you're looking at 2 routes of administration?
We are. We not only have a chance to do an intravenous injection like we did before, but this time, because of the behavior of the new formulation, we're seeing that it's actually quite nicely available by potentially a subcutaneous route of administration, and that's an exciting second possibility. So we're exploring both, and we'll ultimately need to choose a route of administration for our long-term studies.
And that Restore FA study is currently active in Australia.
It is.
I know there's question like, well, when might it be in the US? Are you going to other geographies? The people online are probably in Europe and other places, so.
We are working very hard to expand the footprint of the study. So we have ongoing discussions in numerous geographies across the world currently. But all of these things take longer than certainly I would like, and I'm sure everybody else would like as well. But so you just have to stay tuned for updates on possibilities for other locations. But in the meantime, we're very grateful to have an open study that's active and enrolling, and we're looking forward to gaining more insights into what 216P2 can do.
And this is a study that's ongoing in adults at this point?
That's right.
Yeah. And if people are interested in participating, it does require travel to Australia, but that is, you are willing to accept people into the study who are outside of Australia if they can go and be there for the full term?
We have had a number of people reach out to the investigators in Australia, and they have thus far actually accepted a number of individuals who are willing to travel to their sites.
Great, thank you. I'm going to ask Sandy to go next and give us an update on the Lexeo LX2006 program. Sandy?
Thanks so much. I just want to start by echoing what others have said, which is it's really an honor to be here and have the opportunity to speak with you. And thank you, Jen and Ron, and to all of FARA for putting on this event that enables us to really have an opportunity to share some of the information with you. So my name is Sandy Sitai. I'm the Chief Development Officer at Lexeo. And Lexeo Therapeutics is a clinical stage biotech which is focused on developing gene therapies for rare cardiac diseases. And so we're specifically focused here in FA cardiomyopathy, so the cardiac disease associated with FA. And our approach in terms of developing a new therapy is focused on gene therapy. And so what is gene therapy, and how have we been thinking about this? So as Jen mentioned, and as you all know all too well, is that FA is caused by a genetic mutation that leads to ultimately a deficiency in the frataxin protein. And so with gene therapy, we provide, it's a one-time dose through an intravenous infusion. We provide a functional full-length copy of the frataxin gene that then gets into the body cells, and then the body cells and the machinery can then use that genetic code that we administer for the frataxin gene to then make your own frataxin. So again, as I mentioned, it's a one-time infusion, and we have gone into the clinic. We've dosed 17 patients so far. We recently shared data from our first 16 patients that have at least 6 months of data. We conducted these first 2 studies. We have one study conducted by Lexeo and another that's being conducted at Weill Cornell Medicine. It uses the exact same gene therapy approach and gene therapy product. So when we look at our data and how patients are responding to these therapies, we do pool the data across the two studies. And so across the first 16 patients, we recently released some preliminary data just about a week ago. And so when we designed, one thing just to go back a bit, one thing, when we designed the gene therapy, we really focused on the cardiac disease. And so the reason why we did that is we recognized that cardiac disease is the biggest issue that leads to some limitation in lifespan. And so in doing so, we really focused on measuring the outcomes in the heart. And so when we looked at the first 16 patients, what we're seeing is changes in the thickness of the heart. So in the heart disease associated with FA, the heart walls get really thickened, and overall the heart becomes heavier or bigger. And so we measured through an MRI, so through imaging, we can see how thick the walls are in the heart. And with that, we've seen quite a significant reduction in that heart mass. To the point where for the patients, we have 6 patients who started out with very abnormal thickened heart walls. And when we look at those 6 patients that had that, the heart wall thickness has reduced in size into the normal range for all of those patients. So we've also looked at other cardiac measures, looking at the cell injury that happens in the heart cells. And those markers also are very encouraging and consistent with what we're seeing in terms of the thickness of the heart walls. One other thing that I'd like to mention too is that, through this study, although we were focused on the heart, we certainly did look at neurologic measures throughout our study. And so very preliminary data. We've been very excited and encouraged to see that we are seeing movement on that FARA scores that others have mentioned, the modified Friedreich's Ataxia rating scale. And so we have asked ourselves, well, what's doing that? Because we designed this gene therapy to focus on getting to the heart cells. And so what we did was we looked back at some of our preclinical data, so the data that we have in mice as well as monkeys. And we did see that we were actually affecting cells in skeletal muscle as well as some nerve cells. So we think that as a hypothesis, we're saying maybe this is why we're seeing some changes on that FARA scale. And we do aim to get some data on muscle biopsy so that we can help answer this question, but just feel very encouraged to see movement on scales even beyond the heart.
Sandy, thank you.
Yeah.
We've also heard kind of through, I think, the rare disease community, news of gene therapy trials where there have been positive outcomes lately, but we've also heard about challenges in developing gene therapies and some serious risks associated with them, including people who have died during gene therapy trials. And so it is a very novel treatment approach. It comes with risk. Can you speak to, what's been observed in terms of safety and risk from the patients who've been in, the 16 people who've been in the study so far?
Yes. So in the 16 patients, we look at safety across everyone, so all 17 patients. It has generally been well tolerated. We have had one serious adverse event where a patient did have inflammation in the heart muscle. It happened about a year after therapy. We don't have an exact cause for why a reaction would happen that late, or if in fact it's fully related, but to be on the cautious side, we said maybe it can be, and also seems to be happening on top of what may be just underlying disease progression. And to address the question around, certainly there's been a lot in the news around safety events. I think that the safety profile is often dependent on the type of capsid. So we use a viral vector to transfer the genetic code into the patient's cells. And so the type of capsid that you use can impact the safety profile, but also the dose. And so the dose that we're using here in FA is about 100-fold lower than some of the currently approved gene therapies. And so we think that's contributing to the safety profile that we've been observing with our capsid.
Great. Thanks, Sandy. A few other follow-up questions for you. You mentioned that one of the things you observed when you looked at the heart is that when in individuals who were in the study who had this increased heart mass, that you saw a return of that to normal. What does that mean for cardiac function, or how somebody might feel every day, or what else can you say that might help people interpret or understand what that result means?
Okay, so when we look at heart function, actually most of the patients who came into the study, even though the patients have thickened heart walls, the heart is actually pumping or functioning quite normal. What really matters with looking at the thickness of the heart, what we've seen in terms of some real-world data or natural history studies, is that the thicker the heart is, in fact, the poorer outcomes are for patients in terms of lifespan. And so that's why it's been an important marker or a surrogate endpoint that we talk about in terms of what it may mean for longer-term outcomes.
So Sandy, what comes next?
Yeah, so for us, we currently have an ongoing natural history study. And I know you guys have heard a lot about natural history studies and how important it is. I would say it's ever so important for us who are working in the cardiac space. So I think the natural history data that's been amassed to date across many different studies and have come together, I know that the group has been talking about that, is very strong in terms of the neurologic component of the disease. But actually the natural history data that exists for the cardiac component is just not as robust. And so I know there are ongoing efforts to get some of that, which is more retrospective, meaning looking back at patients' records and trying to pull that information together. What we're doing is a prospective look, so it means that we're able to do these studies on a regular cadence so that we can get more robust evidence in terms of what happens in patients who have the cardiac component of the disease. So that natural history study is open and active. So we have sites open in the US and Canada. We're still opening a number of sites. We do intend to go to Europe and the UK as well as in Latin America. So it's really important. I would just take a minute to make a plug that, whether you're interested in participating in an interventional trial or not, and if you're not, it would be great, if you're willing to contribute your experience if you have cardiac disease, because it does help overall with the development of new therapeutic approaches.
Thanks, Sandy. That study is called Clarity, and there is information on the Clarity study on our trial finder, so thank you. I'm going to ask Matt. Thanks for being patient, Matt. If you would introduce yourself and talk about Solid's gene therapy program that's just getting going.
Sure. I'm Matt Harmelink. I'm the therapeutic head of neuromuscular at Solid. Solid is a biotech company that develops rare genetic therapies for both neuromuscular and cardiovascular diseases. Our drug is SGT-212. It's a drug that we're looking at both for a dual route of delivery, so a systemic delivery, as well as a neurosurgical dentate nuclear infusion. It's, as mentioned before, it is a gene therapy, so there is a viral capsid, transgene. The transgene the idea is to replete or increase the amount of frataxin protein that is typically deficient in someone who has Friedreich's ataxia. We're a little bit earlier along in our development process. We are hopefully going to be dosing very shortly, but have not dosed anyone to date. And so we're starting to move forward in that approach.
Great. So Matt, there was a lot in what you said. This is a gene therapy approach. And let me just tell, we've done 2 webinars on this program. There is a lot involved in this program, so there are 2 webinars where we kind of go through both the rationale for the program as well as what the phase 1 study is going to look like, this first in human study. So it's a gene therapy, and you said 2 routes of administration, one IV and one that is a neurosurgical approach for the dentate nucleus. For the non-neuroscientists, What is the dentate nucleus and why are you injecting there?
So if we start off with just thinking about our nervous system, we have the central nervous system, the peripheral nervous system. The central nervous system is our spine and our brain. In the back of our brain, we have what we call the cerebellum. It's a part of the brain that controls a lot of our coordination and movements. And deep inside the cerebellum, there are these different nuclei that have different functions. On each side, because most of our brain is symmetrical, we have one of those nuclei on each side is called the dentate nucleus. And we know from different studies, literature, pre-animal models, that the dentate nucleus is very involved in patients who have Friedreich's ataxia. And so the idea of our program is to directly target an area that's probably had a high effect of causing disease, and being very targeted in that approach. And that's why we use this neurosurgical approach. So it's not just infusing everywhere. We want to be able to go right to the target tissue that we want to see, inject, and then be able to make sure we're getting the drug to where it wants to go.
Right. And then the IV would assume, like, get to the heart, kind of like the similar rationale to the Lexeo program we just heard about.
Yep, very much so. That's the idea of systemic dosing. We go to the heart and then hopefully affect other organs as well that are affected with FA.
And there's a specific reason why you're doing both at the same time. That is a limitation of gene therapies, but maybe you could just give folks an understanding for why that's important to do these two things together.
Yeah, so as we think about viral vector gene therapies, and this is across the field, anytime we get exposed to some sort of foreign particle, we can form a response. And sometimes that can be a protein response like an allergy. Sometimes it can be an antibody response, for example, to a virus. It's the same premise that people talk about when they talk about vaccination. So naturally in the population, we know some of us already have antibodies to some of these vectors, and it's something that you have to be screened for before you would go into these trials. However, once you get exposed to this, especially exposed to it systemically, you can actually develop an antibody response, which means you would not want to get the drug after. The two reasons are, one is your body already knows it's there and it could actually have a reaction to it, just like if you got another virus and a pretty strong one. But the other problem with it is that that antibody can actually bind the capsid, the virus that you're being infused with, and may make it less effective. The body's working like planned, just not like we want in that case.
Thank you. You mentioned, we talked about the neurosurgical approach. Is this neurosurgical approach something that's new to FA? Has it been used before? What do we know about that?
So the neurosurgeons call this stereotactic neurosurgery. It's newer to FA, but not newer to medicine. Actually, PTC has a drug that they mentioned that has been approved using this. They're going up a little bit higher up than the dentate nucleus, which is in the back. It's been used in other studies in Parkinson's, Alzheimer's. There's a lot of research going on. The same technique has also been used for things like infusing chemotherapy into brain tumors in that area. So the technique itself has been used by the neurosurgeons fairly frequently.
So the phase 1 study that's starting, this first in human study, at the end of the first webinar, you kind of walked us through that it's going to go forward kind of in stages, or what we call cohorts, right? Starting off in adults with FA, and that the first cohort, part of the inclusion criteria is that you're going to enroll people who are non-ambulatory. Then in the second cohort, kind of move to people who are ambulatory. Can you kind of just give any more background on kind of why this kind of study design, and what else might be important to know about kind of key inclusion, exclusion criteria for this study?
Yeah. So we're coming into this first in human study, it's a phase 1b study, meaning that we're looking at that initial safety and dosing in humans. And for that reason, we want to be very thoughtful and sort of walk through as we move forward with the study and the program. The approach to start off with patients who are non-ambulatory is really the idea of being able to treat patients who may be a little bit further along in the disease process. We'll also be able to treat patients who maybe have a little different neurologic functioning as we're looking forward. That's something then that we would expand. The plan is to expand after that first cohort to an ambulatory population and then sort of go from there. Overall, with the program, we want to be very thoughtful as we walk through this. This is a new program. It is also something that is a dual route. So there's a lot of learnings we're going to take as we dose each participant as we go forward, and then come back to the table and make sure we're doing what's best and safest for everyone.
Like we said, the study is just opening now. There are 2 sites listed on clinicaltrials.gov, Ohio State and Children's Hospital of Philadelphia. Is the study actually open? Like, if people call, can coordinators start screening yet, or are we still a few weeks maybe away from that?
It is very close.
Okay, a few days.
Very close.
So we'll keep everyone posted. But it's good time to go back and maybe take a look at those videos and learn a little bit more about this and see what other questions we might have. I also just wanted to mention that one of our educational initiatives this year has been around gene therapies. And so we updated a lot of the information that's on our website about gene therapy clinical trials, and we have a new video coming out as well that we'll be sharing with everyone this week. And so just all resources to try and help you navigate the different trial options and treatments that are being developed. And so with that, I really want to thank all of my panelists for joining us today, sharing information about their programs. And we have a few minutes left. I can open it up to the audience to see what questions I didn't ask that are burning in your minds that you would like to ask any of our panelists. Chandra. She's right here, Ron, right in the middle.
It's not a sciencey question. Is there a name, or is there, sorry, Rusty, is there like a story behind the name Nomlabofus, or the silent P at least?
Well, the original name was unpronounceable, so we had to cut it down a little bit. So the name gets chosen for us. There's a group out there, I'm not going to name them because I don't like them, and you send in what your drug does and how it's constructed and that sort of thing, and they send a name back and they say this is your name. So that's it. I wish I had a better story, I just don't.
Dr. Shaw, for the design therapeutics, what is the timeline for the phase 2? When would you expect to begin that?
So the Restore-FA study is ongoing, and we are looking to initially focus on the frataxin response, and we expect to have data from that sometime in 2026. And then the data from that study will then inform the future development plans. But it's an exciting year ahead for us.
I'm a member of Amelia's Familia, and I'm not antist, but I'm an aunt. No, my question is, has anyone observed changes in your subjects that they themselves can observe and enjoy or not, but just actual changes as opposed to the types of outcomes and changes you've mostly been describing, which are detectable by your own instruments and analysis of DNA, of blood. But is there anything comparable to the young lady who loved riding horses and told us about it today and knew that she was stronger?
Take it, yeah. So thank you for the question. So you've heard about mFARS and a subset of mFARS called upright stability and measuring the thickness of the heart and all of those endpoints that physicians use to measure the course of the disease. But part of our program, and I know part of the program for PTC and for Riata, who developed the Biogen product, also have, for example, patient-reported observations, right? And those things, for example, like they ask about activities of daily living. How is it getting dressed? How is it standing up? How is it eating food? All of these programs incorporate those types of questions. And in fact, we're lucky because thanks to FARA again and the core of neurologists, they've developed a activities of daily living score that is particular to people with Friedreich's ataxia. So we're very lucky to have that. We also have, there are also measures of fatigue that the patients report as well. And we could go on, and actually we have to be very careful. We don't want to overdo it. We don't want to overtax the study with these types of evaluations, but we do have patient-reported outcomes as part of our studies. What are they? Well, the study for Larimar is in progress, so I'm very reluctant to share interim results. As I did mention, they are in the press release. I know that you can share from and Sandy, you can share as well. Yeah, I'm bashful about sharing results when the study's ongoing.
No, the one thing I would add is, you know, the way that we conduct studies, as much as I would love to know and hear about each patient's experience in the studies, we as sponsors often actually don't know and shouldn't know who participates in our studies. So we often don't have the patient level experience other than through the scales that we get. But those scales and scores, as Rusty said, are things that when we look at changes on those measures often tie back to activities like having better coordination to brush one's teeth or get dressed in an easier way. So those are, you know, maybe more tangible things that those scores relate to, but in terms of what exactly we see in the participants in our studies, we actually do stay at arm's length because it's anonymous to us, if that helps.
Hello. I'm a AV engineer myself from Caltech, so I have a quick question for solid bioscience. As we know, when we use AAV to do both brain injection and also the heart transduction, the hope is that we have an optimal AAV for both purposes. But normally it's very hard to have one optimized for both the brain and the heart. So my question is this the same AAV capsid optimized for both, or we are using two different AAV capsid? But if it's not a shareable information, that's okay.
So I don't think we've shared exactly what our capsid is. However, I can say it is the same drug, so it will be the same capsid for both. It is just going to be different doses.
Thank you so much. My name is Ethan. I'm with Matt LaFleur here, really. But I'm also with Friedrich's Ataxia News. We're an online community for patients and caregivers. And we get a lot of feedback in forums and in surveys from patients. And on the one hand, this is great news because we haven't had so much to report on for FA treatment since the site was launched. But we're also getting a lot of questions and confusion about all these possibilities and what to do if you want to go on one trial, but you're worried you might not be eligible for the next trial if you go on that. And I'm just curious if you can give any practical guidance to patients about how to make that choice. And obviously seeing their care provider, but any thoughts you all have for the patients that are here?
Rusty's staring at me like, you're answering this one, not us. I guess that's the genetic counselor. You know, it's the great question, and it's a really, really important one. And I think these are, first off, these are individual decisions. What's right for somebody, for one person, is not necessarily right for somebody else. These are very individual choices based on your medical background, your history, where you are in your FA journey, what you're looking for from a therapy, what your risk tolerance level is. So there isn't one right answer here. The most important things for you to do is get as much information about each of the therapies as possible, and that's why we're doing all the webinars, we're doing the symposiums, we're keeping the website up to date. We're trying to make sure you have as much information as possible. It's important that you have a trusted care provider and somebody who understands these trials and Friedreich's ataxia to help navigate you through that as well. You should be talking to the study investigators and asking them questions and letting them help guide you in this process. It is a lot of information, and it's changing almost all the time, right? A year ago, this was a different panel discussion. It could be different 3 months from now, the information that we have available to us. So even the information available to you now might be different 6 months down the road. So, you know, it's important to have a trusted care partner who you can talk to, who can help work through the pros and cons. Again, we've got the clinical trial guide that tries to take you through some of the key questions you should be asking yourself about participation in trials. But, you know, being informed, having someone who's trusted, who can help guide you through the process, and supporting each other in the decisions that folks make is also important. Knowing that there isn't just one choice for everyone would be my guidance there. All right, one question online.
I'll try to combine a few of the questions that came through Zoom. I think we can fit them together. One of them was about whether you can receive a second gene therapy after receiving one, which I think the panel has already shared that that's not possible because you develop antibodies to the gene therapy the first time you receive it, to the viral capsid. The second question is kind of related to that. Someone was trying to understand for Lexeo whether you have any preclinical data that shows that your gene therapy does reach the central nervous system, or are you just looking at the FARA scores that you're collecting in the patients?
So thanks for the question. So we don't have any evidence that we get to the central nervous system. We administer the drug peripherally. But we are doing the mFARS to, like you said, to see if we are getting neurologic benefits. Yeah. Yes, and to the first part of the question, which was around the gene therapy administration. Today, it seems that you can only do one-time administration of gene therapy. What's not clear is if they're different capsids, if one would be able to tolerate one and not the other. Not enough is known about that yet. There's a lot of research ongoing that is looking into whether you can re-administer gene therapy, but that is not where we are today. So for now, you know, as people make decisions, I would make the assumption that it is one-time gene therapy for the moment. Yeah.
All right. Well, thank you to our panel.